All right, again, welcome everybody to the second session of Breaking the Ice with APECS-Chile. We're really excited on this very hot Wednesday here, actually, where I'm at right now. But we're ready to talk about cold stuff, so cold and cool stuff. And I wanna tell you guys some instructions that we have for today in order to go smoothly with the presentation. We are taping this and it will be available on our channels and also on YouTube in case you have friends that haven't been able to see this presentation. But you're also not allowed to tape it. So please respect that, and we will keep your microphones off, but whenever you have a question that you believe, do you think that is easier for you to speak it out loud, you can raise your hand and we will we will activate your microphone right away. This is a conversation with our guests, so you can actually make a couple of questions along the presentation as well. He doesn't have a problem with that. But you can also leave your questions on our Q&A section and we will read them for you in case you don't want to speak. We're first going to start thanking. There is a whole team behind this. There is, first of all, the people from the Polar Week group of APEC International that has been helping us a lot with this organization. But we are APEC-Chile, and specifically APEC-Chile is run by a directory. So Claudia Maturana, Estrella Alcamán, Sandra Lopez, and myself, Renato Borrás. I'm sorry, yeah, I'm Renato Borrás, the national representative of APEC-Chile. And we also have some people that works in the design of our website and the flyers, so we're really thankful with them too, Alejandra Ramirez and Constanza Rojas. Finally, we have a lot of people that has been collaborating in spreading the words around. First of all, Red Lama from Center of Climate and Resilience Research. We have the GAP, which is the Genomics Antarctic Biodiversity Group. And we also have the Chilean Antarctic Institute that recently released a note about Polar Week as well. So you guys are more than welcome to read those. Yeah, the next session that we have, we're gonna have Dr. Julieta Orlando tomorrow. We're also gonna be talking about microbes, and this is actually, the title of the talk make a reference to one very famous Spanish song, so it's really hard to translate because that talk is going to be in Spanish as opposed to the talk that we had on Monday and the talk we're having today. So there is really no roads on science. You will make your own road on your own. So it's the more of a review of the story of Dr. Julieta and around her area of expertise, which is microbiology, and what she has been doing in lakes or the ocean and Antarctic soils as well. So that's going to be tomorrow. There is also, don't forget that you need to register for this. Don't forget that this is in Spanish, and look out for the link for registration. You can send us an email or find those on our social media as well, and also the link of our website. That being said, I'm gonna start with this. I'm really excited. Today we have another brilliant Chilean guest, and I'm thrilled to present Dr. Eduardo Castro Nayar. He's an associate professor of the Center of Bioinformatics and Integrative Biology of the Universidad Andrés Bello here in Chile. Eduardo obtained his bachelor degree in biochemistry at the Universidad de Santiago in Chile, and his PhD in biological science at the George Washington University in the US. Actually, he is also an adjunct researcher at the Computational Biology Institute on that same university in the US. His research focuses on experimental and computational techniques to understand the causes and consequences of microbial genetic diversity in natural, built, and host-associated environments. Currently, Eduardo is studying microbiology communities in environmental systems, aquatic, terrestrial, or aerial, and host-associate, which is humans and plants systems as well. All these projects are related to extremophiles and limit of life biology, such as places of Antarctica, which we're going to be talking about today, but also Patagonia fjords and the Atacama Desert. He's a fellow of the UC Davis Chile program as well. And I think Eduardo has been in Antarctica already twice, and where we actually met a couple of years ago. And just recently, he got his first grant leading a project in Antarctica. And he will be talking about all of that. And we as APECS-Chile are very happy and very proud to have him with us today. So Eduardo, I'll give you, you can now start sharing your screen, and we'll go with you. And thank you very much for being with us.
Excellent, thank you, Renato. Let me see if this works. Perfect. So thank you very much for the introduction. I'm really honored to be here, to have been given this possibility of showing you our work. As Renato said, I'm part of the Universidad Andrés Bello here in Santiago, and I'm leading a really multidisciplinary group in Antarctic research called Polarics that I would like to talk about. This is a rather unusual presentation for the simple reason that we don't have many results to share right now. You know, we got the project, the grant funded last year, December last year, and then a pandemic hit us, and here we are. So I'm going to show you a little bit of the work we've been doing in Antarctica for the last few years, and then I'm going to move to the Polarics project specifically. Right. If you wanna more information about our work, you can visit this website, or you can find, you know, what the members are of the lab and our current research and things like that. So I'm not really an Antarctic researcher by birth, but I'm one by choice, right? So I was trained as a biochemistry, although I've never done any biochemistry. And then I went on for my PhD in the US under the direction of Keith Crandall, first at Brigham Young University and then at George Washington University. And you know, I was lucky enough to be in the what I call now the microbiome fever. You know, everything was microbiome when I was doing my PhD. We had new sequencers and new techniques, new bioinformatic methods, and I landed in a computational biology institute. So from biochemistry to computational biology and the microbiome fever, that was essentially my thesis, my dissertation, and we contributed a lot of things in the method landscape with this piece of software, a fairly famous piece of software called Pathoscope, and we also got into a new model, which is the upper respiratory tract, which is really interesting actually, especially when you look at disease and healthy people, especially in the asthma setting. That finished by 2014, and in 2015 I returned to Chile to start my own lab. So the upper respiratory tract is really nice because you could say there are many environmental gradients. Or physiological gradients. As you go from the nasal cavity, you have certain diameter of particles that can enter the system, right? And this is actually inversely correlated as you go along, right? pH is inversely correlated with that. And you start at 6.3, and you can move down to 7.5, so almost 1 order of magnitude. And the surface, of course, is it starts off very limited, and then once you get to the lungs, this expands greatly, right? There are many physical-chemical gradients, if you want to. Pressure of oxygen, pressure of CO2, relative humidity, and those things are really nice to study from the microbial perspective, because there is a bunch of environmental filtering going on here, and environmental selection, if you will, right? But that was human, right? I wanted to reinvent myself when I started the lab, and taking probably, the best experiences in the PhD, the training, the people I met and all that, but moved to the environmental landscape, really. So in the lab from 2015 to now, we have started at least 4 different projects. We have been studying some bacterial genus in the Atacama Desert in the north, even whales off the coast of the Pacific coast of Chile, aquaculture effects on fjords in microbial communities in the south, and also these endophytic microbial communities of these very picture-ready, I would say, a tree called monkey puzzle tree or Araucaria in Chile, trying to look for pathogens and understanding a little bit about the microbial communities that inhabit this tree. So all these have taken me to many places in Chile, in this really long country with a lot of environmental gradients and different climates and different landscapes. But there was another frontier, right? The next frontier for me and for my group was, what about Antarctica, right? And Antarctica really is unparalleled in terms of conditions. It's the coldest, driest, and highest continent on Earth. It's mostly pristine, although that's somewhat controversial. And it's been ecologically isolated for possibly millions of years. And that kind of, it's also controversial, right? There's a lot of life going on in Antarctica, especially in the coastal areas and in the ocean. But everywhere you look in Antarctica, even in the peninsula or the Antarctic plateau, you can find that Antarctica is microbially rich. So microbes are really the kings in Antarctica. And that was really interesting for us. So microbes in Antarctica have to deal with a lot of new things, right? Things that you commonly as a microbe don't face in other continents, right? And Antarctica has many different environments. So of course the ocean is probably one of the most well-studied environments, but you also have subglacial lakes, like here, where you find a lot of microbes that can use methane, for example, the gas methane as food, really, as carbon source. You have really cool metabolic pathways going on in glaciers and in the soil, and you even have microbes that do not use light to create their own food. They need carbon from other sources, but they're really adapted to extreme oligotrophy, right? So there's not a whole lot of organic carbon in Antarctica besides the coastal areas. And microbes have learned and adapted to use the few sources of carbon even within rocks, like in this example. And we have a lot of different lifestyles for microbes in Antarctica. We have acidophiles, so that would be microbes that like acid environments. Alkaliphiles, the opposite, right? Alkaline environments. Allophiles, they like to live in salty environments. Thermophiles, and that would be microbes that really like hot environments, which is kind of a counterintuitive, right? You think of Antarctica, you think about ice, you think about cold, right? But there's a lot of thermal activity going on in Antarctica. There are many active volcanoes and thermal vents, and in those places, microbes that love hot environments really thrive. And you even have hyperthermophiles, which are microbes that can live above 50 degrees Celsius, right? Cycrophiles would be, and cycrotolerants would be some of the most common microbes you can find in Antarctica, because these are cold-loving microbes, and you also have cold-tolerant microbes. They don't really like to be in the cold, but they can grow anyways. Okay, so really my experience in Antarctica starts here in these places, in the Shetland Islands, specifically in these 3 islands, in King George, in Greenwich, and in Deception Island, where I started collaborating with other researchers that took soil samples from here, right, from all of these places. And we looked for particular microbes, right? Most of the microbes that you can isolate in Antarctica are actually microbes that you can find in some other places in the world. In this case, I'm showing you some results from Pseudomonas, right? It's a really ubiquitous microbe. You can find Pseudomonas nearly everywhere in the world, especially in soil. And Antarctica is no exception. What is really cool about the microbes that you can find in these places is that they have traits that are really special, right? For example, we have found Pseudomonas species that can live in presence of telluride, a really toxic metalloid, right? Some even show cross resistance, like they can if they have resistance to one metalloid, they can live in the presence of another metalloid. There are things that are a little bit off also when you look at the metabolic potential of these microbes. For example, most of them are resistant to antibiotics, even antibiotics that are used in the clinic, right? Human class antibiotics like streptomycin, chloramphenicol, and kanamycin, and we have even experimentally confirmed this. And you also have microbes that we've isolated in these places that are even capable of degrading TNT, the explosive, right, by biotransformation and reduction of the nitro groups, right? And it's not just some sort of a cross resistance, or you could say that maybe the genetic machinery of these microbes is being co-selected for something else, right? Oftentimes you see genes that can work under one scenario, and the same gene can work for a different situation. Most of the microbes that you can isolate actually from these islands have a specific genetic machinery that allow them to perform these functions. And when you look all of these in a phylogenetic perspective, they don't always cluster or form clades with other Antarctic microbes. So it seems that they are there, but they are either recent migrants or they really do not belong there, right? This one is really interesting because Pseudomonas MPC6 is resistant to arsenic, right? Very toxic to humans, and cadmium, also very toxic. But it's also resistant to vancomycin, which is a really last resort antibiotic that we use in the clinic, right? So why would Pseudomonas from Deception Island would be resistant to vancomycin, right? It's surprising the range, the expand of phenotypes that these microbes can exhibit. Some of them can synthesize biopolymers, and they are really versatile in terms of temperature. For example, this one, MPC6, can grow from -5 to 45 degrees Celsius. And it's interesting also because its optimal growth temperature is not even at a temperature that is relevant for Antarctica, right? The optimal here for MPC6 is 25 degrees Celsius, which is weird because that you never find that temperature in Antarctica, right? So the message that I'm trying to put across is that many of these microbes seem to be cosmopolitan, at least from the taxonomic point of view, right? Most of them are cyclotolerant and not cyclophilic, and that means that they can grow in really cold conditions, but they don't really like it, right? They would rather grow at 25 Celsius degrees, for example, right? And they often exhibit traits that allow them to deal with compounds, with chemical compounds that are not necessarily from Antarctica. So we could say alloctonous compounds. Then in my group we moved to understand more the community ecology side of the microbes in the soil, and for this we got interested in plant microbiota interactions, because plants are not isolated, right? As we historically, I would say, been studying plants, right? Plants are part of a community, and they are really in tune with the microbes that surround it, right? And there are some expectations. For example, the soil microbiome or the soil microbiota, it's normally or regularly is really diverse in terms of alpha diversity, and then you see some gradient of diversity as you move along these plant modules, where you can distinctly define the phylosphere. That would be the microbial communities from the leaves, the air microbiome that interacts with this phylosphere, and the root endosphere, for example, and all of these. And it's not simply that microbes are there in the surface or in between the plant tissues. They are functionally related, and normally these microbial communities offer ecological services to the plant and vice versa. So it's really an intimate interaction. And you can even expand this holobiont concept, the plant microbiota interactions, by adding up the ecosystem, right? So in this picture, these authors, is a figure from a paper recently published in 2020, and the authors essentially expanded this framework to talk about the microbial cycle and how different animals interact with the soil microbiome. The soil microbiome in turns provides ecological services to the plants and so on. With that in mind, we started working on the Borrás Peninsula in a collaboration with Marco Molina and Jorge Gallardo from Universidad de Talca in the central south part of Chile, specifically looking at these two plant species, Colobanthus quitensis and the Da+Ca antarctica. These are the only two vascular plants present in Antarctica, native plants from Antarctica. And we performed the first metagenomic study using shotgun sequencing on only 6 samples, a rather preliminary study. And we found that when you look at the rhizosphere of Colobanthus and you compare that to Colobanthus and the Da+Ca, you can see subtle differences that no matter how subtle they are, you can find significant differences, right? So these essentially hinted at us that the microbial communities are being selected, plant selected, right? And this is all related to the theory behind the rhizosphere composition, whether the plant species through plant exudates or root exudates can select microbes from the soil, recruit them, if you will, and use them as partners, right? Or the rhizosphere is really defined by whatever it was on the soil, right? And we see some specificity when we look at these microbial communities in the root. And after that, you know, for me personally was the right time to go to Antarctica. You know, I've been working in Antarctica for a year or so with Antarctic samples, but never been there. So thanks to our collaborations, I went to Antarctica for the first time. My experience was the Borrás Peninsula in Livingston Island. And of course, the first thing I did once I got to Antarctica was to take a picture of penguins, right? As anybody should do when they got there. Of course, I was amazed by the diverse landscapes you can see. My impression or my preconception was that Antarctica would be essentially a block of ice, right, with not a whole lot of diversity. But that couldn't be farther from the truth. I saw a lot of diverse landscapes and really nice sunny days and a lot of fauna and life, really. So in order to get to the Borrás Peninsula, you had to go in a Navy ship like this one, and once you approach the peninsula, you use a helicopter to get there. And essentially, once you are in the Borrás Peninsula, you need to set up camp. These are our personal tents. This is like the dining area, if you will, the common area. You set up the sampling trips from here. You eat here, and you talk to other people here. And this is kind of your private space and the places where you sleep. And of course, once you're camping out in Antarctica, you have to take a leave no trace policy, right? Nothing can be left in Antarctica. So even our you know, our organic rests, right? And we have a bathroom or a restroom tent as well. This is essentially your food, you know, a lot of granola things and coffee and dehydrated food. There were many long walks to get to the sampling sites until finally found some colonies of Colobantus, which is somewhat scarce in the area. The Champs Antarctica, the grass is really more ubiquitous in the island. And these are some of the pictures of our team working here in this slope, taking some samples from Colobantus. Of course, Jorge here taking some notes, and Flo and myself are taking some samples in the back. We even took some reference soil samples from this glacier. It's a glacier that almost makes it in this area, at least, to the ocean. And there's a small strip of land. You can call it a beach or some sandy area, coarse grained, and we took also some soil samples from there. Then helicopter back to the Navy ship, and more beautiful landscapes. This is actually not a sunset; it's a sunrise, but it really looks like a sunset. And then back to the base, to Professor Escudero base in King George Island. So we took a bunch of samples, over 100 samples, and for those that are not familiar with the place, the Livingston Island is here. It's part of the South Shetland Islands here in this archipelago. Specifically in this region. So you normally would come from Chile or Argentina, and then land in King George Island, and then move in this you go island hopping until you go to the place where you're gonna take the samples. In our case, we set up camp over here, and we had 6 points of interest where we knew there were plants or plant colonies, right? And we took samples of the Antarctic grass, the Champs Antarctica right here, of Colobanthus quetensis, this one, Antarctic pearlwort is known in English. And we also observed that there was this particular association where you literally have some Colobanthus quetensis living in or on top, if you will, of this patch of Antarctic grass, right? So we were really interested in learning about the rhizosphere of this system. And of course, we took bulk soil from really close to the plants to compare, right, to have a reference. So the first thing we did was take a look at alpha diversity. There were some predictions in terms of, we expected a diversity gradient from the roots or from the bulk soil being the highest diversity sample to the roots or to the soil surrounding the roots and the rhizosphere, right? And we tend to see this in these panels. I'm showing you 3 different types of alpha diversity measures. This is CHA1 richness index. This is the Shannon's diversity index. And this is Faith's phylogenetic diversity. And we have 3 colors in the graphs. Rhizosphere, that would be in green. Then the soil that is surrounding the rhizosphere, but is not in direct contact with the roots. And then bulk soil, that would be soil apparently not, or seemingly not under the influence of the plants. And we kind of see this, for example, if we look at the Faith's phylogenetic diversity, we see that the green, they all overlap, so they are not significantly different necessarily. But the green section is here, so rhizosphere is really low diversity in comparison with the soil surrounding the rhizosphere and the bulk soil, which are over here. So that was the first thing we learned from this system, and the obvious questions for us were, is it the soil or the plants, right? What really drives the community structure of these microbial communities? right? Is it the plants that are selecting the microbes, or they're taking whatever is in the soil, right? So we did this RDA analysis to interrogate the data in terms of what's the explanatory power of the sampling sites, right? If you constrain this multidimensional scaling by location or sampling sites, what do we see, right? And we see that most of the variance, the diversity observed, 55 plus 17 in this case, is explained by the sampling site, with some sites that are more diverse than others, right? And this is true for bacteria, which will be panel A, and for fungi, which is panel B, right? So it seems that the main driver of the microbial community structure really is the sampling site. It's whatever microbes there are in the soil, right? And then, of course, we looked at the composition, kind of answering the who's there question. And we found a number of different microbes, some known, some unknown, some classic members of the Rhizosphere that you could find in any other continent, and then also some specifics from polar regions, right? In different abundances, relative abundances. So the other question was, can we source track the plant's rhizosphere, right? What's the origin of the rhizosphere? How do the plants acquire the rhizosphere? So we did a sink source analysis where the sink is the rhizosphere from one of the points, sampling points, which is called Devil's Point, and then the source could be any microbe from bulk soil, from the soil surrounding the rhizosphere, and the rhizosphere of all of the sites. And we have a panel A for bacteria and a panel B for fungi. And in bacteria, we see that most of the abundance that we see in the rhizosphere from Devil's Point is accounted for microbes that are already present in some other parts of the peninsula, right? So that was indicative for us that maybe the bacterial communities are being selected by the plants, because essentially they select the same thing from everywhere, right? In turn, when you look at the fungal analysis, you see something different. You see a lot of gray here. These bars in gray indicate that the source is probably unknown, right? So in some cases, for example, in this Colobanthus plant, most of the rhizosphere in this case comes from the rhizosphere of some other Colobanthus plant. So it's really similar, right? But it's not always the case. In most of the cases, I would say the source of the fungal communities from the rhizosphere is unknown, right? So with all this, I want to say
before.
Yes.
Oh yeah, go ahead. No, finish your point. I have a couple of questions before we continue.
Okay.
Finish your point with this.
Okay. So I just wanted to say that it seemed to us that the overall composition of the soil, it's made up from cosmopolitan microbes, right? So microbes that you could find in some other places. Taxonomy in this sense, taxonomy alone is not very predictive of the origin of the microbes. We can find Pseudomonas. We can find some microbes that are classic microbes that you find in the roots of plants, but nothing really specific, at least when you look at the taxonomy, right? And of course, there is a caveat, and the caveat is that there are many unknown taxa, and this points a little bit to the analysis that we use, that of course you are not able to put a name, a label on something that's never been seen before. So, Renato, you have any questions?
Oh, yeah.
Yeah,
well, if we think about it, not everything is everywhere. So, and you showed a bunch of different places where you were taking samples, not just one of the islands at the very beginning. So one of the questions that we have here is that despite the ubiquity of the bacterial species, do you think is there a biogeographic patterns within the Antarctic continent?
I think so.
Like some,
I think so.
Yeah.
So I agree with that. There is a null hypothesis in microbial ecology, right? That the Da+Ca hypothesis that says something like everywhere, everything is everywhere, but the environment selects, right? And I think that was normally accepted until recently, but now we come to realize that there are biogeographic patterns, historical patterns. There is a migration, and that's not random. And I think you can see this. Maybe not over the gradients that we've been looking in Antarctica so far. But when you compare different studies, it's very clear that you have a combination of endemic microbes that probably been there for many years, and then also some alloptonous microbes that are probably newcomers to the environments.
Okay. And the other question is it's related more to the paper that you were showing about the Rhizosphere microbiota, mm-hmm. because we had a few weeks ago somebody, also a microbiologist, that was telling us how the animals' depositions can affect the soil community in multiple places, and depending on the animal that was which from the deposition was coming from. You guys in that paper, I think that's the paper that you guys published last year. You didn't really find differences on the physical chemical characteristics of the soil when they were related to each plant. There was only differences in the gene composition, and therefore also the rhizospheric the microbiota itself. I just want to kind of engage to your question that you were addressing right now of whether the plant choose the bacteria or they just get what they have there. And is that something that has been defined? Is there an influence, even though you're not finding physical chemical characteristics change? Is there something that this sort of new genetic composition found on the plants can give to the soil itself?
Right. So there are two sides to that. One is that if you only assume that the potential origin of the microbes from the rhizosphere of these plants is soil, then you are overlooking a lot of different other sources, right? Especially because these plants tend to grow in coastal areas, and coastal areas in these islands are highly populated by birds and also by marine mammals, right? Some of the colonies even are right next to elephant seal colonies, right? So there is an input of organic matter and microbes from these animals. That if you want to look at the origin, you need to consider those. So, for example, for the fungal component that we were not really sure what's the origin, it looks like it's not really soil. It very well might be animals, elephant seal poop, or things like that.
Yeah.
And the other question was about, I forgot.
No, it was whether there was another influence beside the physical chemical characteristics that could occur, right. Like we were seeing in the other results that there right. was I might not formulating this right, but we're leaving a genetic composition in the soil behind after an animal is pooping. Is there right. something like that when it comes to plants? Like
right. You mean like right? I think there might be some vertical transmission as well of microbes. So, for example, right. if you look at the root endosphere of plants, many times is vertically transmitted. So from mother to child, if you will, right? And the other one is that I think there is a whole lot of historical contingency going on in the soil. Like different soils have developed different microbial communities, and there might be a combination, a mixture of the plant species selecting some of the microbes, but they have to make do with whatever is in the soil, right? So right. that yeah. kind of creates these patterns of these structures, right? It's a guess. I don't have much evidence for that. So.
Yeah. We have
Oh,
one more, and then we'll move forward, and we'll save another one for later. Cosmopolitan soil microbes that you see are at higher taxonomy. Could you see the same at lower taxonomy when it comes, like when we talked about a species, for example?
right. So with the amplicon sequencing that we do, we are only confident at the species, at the genus level, and our classification rate for genus level might be around 50%, something like that, with the best methods that are out there. So I could only speak about genus or genera, different genera at that level, and it seems to be a mixture, right? So you have a lot of Cycrobacter, for example. That you know, the name kind of tells you that it's a bacteria that is adapted to cold environments. But you could also find Cycrobacter in associated to hosts, to invertebrates, marine invertebrates, for example, seals and some other animals in different parts of the world, in the Arctic and the Antarctic. So yeah, they show some specific adaptations, but of course, Antarctica is not the only cold place on Earth, right? So you know, they look like cosmopolitan in that regard. A lot of Pseudomonas. I have to say that when you do culture-dependent isolation of these microbes, oftentimes you tend to recover the usual suspects from soil, Burkholderia, Pseudomonas, and things like that. And I believe it's just because we use the media, right, that are being developed over decades for these very same microbes from other places. So it's kind of a bias assessment of what's there. When you look at the metagenomics or amplicon sequencing, you can see something more diverse, and some of them look very particular, and even some we can't put a label on them. So those are highly suspects for endemic microbes. So.
Okay.
Cool.
Yeah.
So
let's keep going.
Okay. So one of the questions we had going forward to, in the formulation of this project was, do microbes migrate to and from Antarctica, right? Or are they endemic? We're by far hardly the first ones to ask these questions, and people have been working since the 60s and 70s on this type of questions in Antarctica. But we wanted to know and fill some gaps in terms of the relationship between South America and the Antarctic Peninsula, right? And one of the first things that came to our attention was this paper published in 2018 by an international group, including Chilean scientists, where they look at Durvillea antarctica, this algae that they found in King George Island, right? The accepted natural range or distribution for this algae is actually indicated in this figure, in this map, with the white triangles, right? So a number of sites in New Zealand and the Pacific coast of South America, the Malvinas or Falkland Islands right here, but not within the Antarctic continent, right? So the first approach of these researchers was looking at population genetics, right? Looking at SNPs, genome-wide SNPs of Durvillea antarctica collected from different places, and trying to build some sort of a SNP tree, and in that way seeing whether, or trying to infer rather, the origin of this algae that they found here, which are pictured here. And amazingly enough, they found that the King George Island algae belong to or form a group with the Kerguelen, which is over here, right? So really, really far away, and somewhat outside the influences of the circumpolar current or the Antarctic front. So they actually speculate that this might be a storm force. So this algae could actually travel long distances, long range travel, aided by storms, and without any need for human introduction or intervention, right? And possibly global warming would be increasing the rate of colonization, if you will, of these plants into the continent, right? They also gather evidence from oceanographic models, and I took this from the publication. These are some 140 particles that they simulated traveling in a 2 years time period from outside, I guess, the effect of the polar currents, and we see that some drift north, the ones in blue, and some the orange particles go back to the Antarctic continent. So really reaching Antarctica from the sub-Antarctic is possible without any need of human aid, right? So our first thinking in the face of this project was, well, is this true for microbes or not, right? Or what could be the transport ways from outside the Antarctic continent to Antarctica, right? And is it water for microbes? Is it air or even animals? And it seems to be all of the above, at least possibly, right? There is a lot of evidence from dust particles and organic pollutants for atmospheric transport. There are some biological pumps, certainly biomagnification is going on here, and hydrospheric transport is also possible, right? What about teleconnections between Antarctica and South America? We were particularly interested in those because we felt that they were not addressed properly in the current literature. And we found that effectively dust particles from Patagonia can reach Antarctica in about 2 days, right? Most of you can see here, most of the particles do not go to the west part of the peninsula, but rather to the eastern part of the peninsula. From all of the particles, from the totality of the particles that can move from these areas, the San Juan area here and northern Patagonia here, about 25% of the dust particles reach the western side of Antarctica. So it's totally possible for microbes to be hitchhiking dust particles and then traveling from Patagonia to Antarctica, right? And in a most comprehensive study from looking at data from the 70s to today, or 2015 rather, researchers have found that the peninsula is mostly connected to South America. That would be the red dots over here, and the purple dots indicate the connection between New Zealand and the Antarctic Peninsula, right? Especially this section. So there is evidence, ample evidence from those particles transport teleconnections between different parts of the world, and especially Patagonia to the Antarctic Peninsula. And there is also evidence of long-range transport of pollutants, organic pollutants that can reach the peninsula from the South Atlantic and other places, especially the western side of the peninsula. Is there any evidence of long-range transport of microbes in other parts of the world? That's a resounding yes. There is ample literature documenting transport from Europe to Africa, Africa to South America, North America to Europe. Emilio Casamayor's group in Catalonia, in Barcelona, published this paper in 2018 showing a long-term survey of dust particles and microbes that come from different parts of the world, right? They have an observatory in the Central Pyrenees over here, and they would able to define the origin or the provenances of these dust particles and microbes that fly through there from either North America, the Atlantic, some regional inputs over here. North Africa is a very good source of dust particles and microbes as well, and the northern part of Europe as well. So it is documented, right? In that regard, we have evidence of long-range transport of dust and pollutants to Antarctica from South America, and microbial transport in many parts of the world. We came up with Polarix. That's the name of the project. And that's really the short name of the project, but because the full name is what you see here. It's long-range transport of xenobiotics and microbes, teleconnections and influence on terrestrial ecosystems. So what we really want to do here is understand the teleconnections between South America or Patagonia in specific to the Antarctic Peninsula through the lens of the potential pollutant and microbial transport, right? And for that, we have defined many areas of research that I'm going to show you later on. But essentially, we want to evaluate reservoirs, possible sources, and potential impact on terrestrial biota. So terrestrial biota is really a big word, right? It's just too much for a single project to address. So we're focusing on the plants, right? Given the expertise of the team, we're focusing on what could be the effect of microbes, microbial deposition and pollutants deposition on the plants, on the soil and the rhizosphere. Okay, so the National Antarctic Institute, UNAB, provides you with a platform, right? Either if you are Chilean or an international researcher, you can apply to support from UNAB. You could get funded through some other institution and then collaborate with UNAB so that you get access to the Antarctic Peninsula, to the bases, refuges, and scientific stations that UNAB manages in the area. And that's what we are actually using for this project. So originally we were interested in some of the islands of the peninsula here. King George in Admiralty Bay, that's really attractive for us, as well as the Byers Peninsula. Greenwich Island could be an alternative as well. But we wanna be rather comprehensive in terms of catching air and microbes from different sources. So we also want to go south to almost the Antarctic Plateau here in the Union Glacier. We're really looking forward to take samples from here. Of course, there are no plants here. It's mostly soil and ice. There are reports of microbial life here, of course, but we're really interested in the airborne microbes that we could collect from these sites. And to get there, you can hop on the Achilles Navy ship right here, or you can take the Hercules aircraft to the peninsula. And the entrance point for INACH, the National Antarctic Institute, is King George Island in Chile Bay, and then you move from here to different places of the peninsula. Okay, so one of the areas of the Polarix project is microbial biogeography and population genetics. We want to look at biodiversity in different compartments in air and dry deposition, soil, rhizosphere, and we really want to tackle some of these questions, approaching using high throughput sequencing and bioinformatics and that type of techniques to assess whether we see to what extent really endemism versus non-native microbes. There are really few studies of airborne microbes in Antarctica. This is a paper from 2016, and they detail the places and the number of sites in these places in the whole of the Antarctic continent, where studies using airborne, not only microbes, but aerobiology really have been conducted. And you can see in the McMurdo dry valleys and this area, there is a number of studies. There's one here in Adelie Island, I believe, and some others over here. But really, there's not much going on in the peninsula, and thus far, nobody has tried to answer whether there is some sort of a long-range transport of microbes and pollutants from South America, Patagonia to Antarctica. So there are some new exciting developments, and starting from acknowledging the importance of the atmospheric microbiome and whether to what extent we impact the atmospheric microbiome. And there is certainly global scale aeolian dispersal that has been reported in certain communities in Antarctica, especially in Western Antarctica and this side, and some other very high profile papers that have been really an inspiration for our project. We also want to look at the atmospheric deposition and soil levels of organic pollutants. Here, the main question is understanding the relative contribution of local and global transport, and that would be atmospheric, of course. And we're probably going to look as well as some hydrospheric transport into the peninsula. And for that, we need to, or in order to integrate all of these results and data points, if you will, we need a bigger model, right? So part of the team are people that work specifically in climate and meteorology and modeling climate. And we want to know whether, first of all, what are the major pathways to Antarctica? Are the New Zealand and South America pathways, to what extent those are relevant to the Antarctic Peninsula? And in a changing climate, right? What, according to the IPCC predictions, what we should expect to happen in terms of microbial or particle transport, right, from these places. We not only want to look at the microbial communities, but we also want to look inside the microbial cells, and we're working on artificial laboratory evolution experiments, trying to push artificially mesophiles, microbes that live at human temperature, so to speak, right? Push them to temperatures and conditions that are relevant to the Antarctic continent, right? Can we do that, and can they probably or possibly maintain their phenotypes, right? There are many microbes that are able to degrade or to use as a carbon source many of these organic pollutants, right? Can we push some of those microbes to adapt in the lab to conditions that are relevant in Antarctica, right? I mentioned briefly that if you use culture, you're probably gonna bias whatever you find, right? Oftentimes you don't even culture the most abundant microbe in a community, but the easiest to grow in lab conditions, right? So we're gonna use this type of device, the isolation chip, or iChip for short, which is essentially is a sandwich of membranes where you put some microbial dilution, a suspension, very diluted suspension, and then you wait many times by submerging. You submerge this device into the soil or whatever it is that it's the original environment where you're trying to isolate these microbes. This type of device has been used in the past. It was first described in 2010, so it's a decade-old device, and it's been used even for antibiotic discovery, right? So new classes of antibiotics have been discovered by accessing the microbial dark matter, right? That huge fraction of microbes that we know exist but we can't culture, right? And so members of the team managed to reproduce this using surgical steel right here. It's a perfect copy of the original, and we're already trying to isolate microbes from air and from soil. And these ones actually are from air. We did some preliminary experiments, and you see a number of colonies here in two types of media, in rich media without any selection, just a nice place to grow for the microbes. And after 5 days approximately, you can see colonies growing, and we're in the process of identifying those colonies. And the final area of research of this project has to do with the impact of organic pollutants on the plants and their associated rhizosphere. So for this, we're gonna not only look at the plant's physiology, but also genetics. So we want to do some sort of a multi-scale approach to try to evaluate the plant's responses to temperature, humidity, and looking at DNA damage, gene expression, membrane stability, and things like that. And we're going to start this probably in the next austral summer. So as I said at the beginning, we're not under this project. We haven't been to Antarctica yet. This was granted in December last year, and our first fieldwork is scheduled for the next austral summer. So the National Antarctic Institute is probably going to take us there in December or January, right? With all the precautions for.
Eduardo?
Mm-hmm. Yeah.
Exactly, with all the precautions of coronavirus, which is another question
I wanted to say, yes.
Well, yeah, we're gonna have to talk about it. Yeah, first of all, and this is kind of tied with the coronavirus question that we're gonna tell you about. You have been talking about transfer of microorganisms from the continent by all these teleconnections, but there is a different thing, and this is more an ecological perspective, which is the establishment of these colonies on Antarctica. How do you evaluate if they can, because one of the things is just reach Antarctica and the other one is just to actually be there and be part of the ecosystem itself?
Right. No, that's an excellent question, because there are many studies, for example, done in Antarctica that they look at soil and different matrices in Antarctica, and they see, for example, pollen grains from species that would never grow in Antarctica, right? So one thing is being transported and deposited, I guess, on the soil, right? But the next thing is, are you thriving, right? Are you active, right? And for that, we use a combination of not only DNA sequencing, but also RNA sequencing, right? So one of the first things we want to do is, from a sample from the air, being able to determine a taxonomic profile of microbes, if you will, by looking at the DNA that would tell us what's there, right? And the same picture, but with RNA, and that would tell us which ones are active, right? Actively transcribing genes, right? Yeah. And we're planning on doing the same for soil and for rhizosphere. So it's a really good question, because you could be fooled by thinking that if you see a DNA molecule, you're able to identify or classify that molecule into some sort of a taxa, right, or taxon. It means it's there, right? And it really doesn't mean that, right? It means that it reached the place, but probably not active, right? Yeah. There's a whole lot of environmental filtering.
Which is exactly what happened with the right. Which is exactly what happened with the algae of the work of the algae were not really there. They were just there's been, I talked with one of the guys, another guy that's been going there for 60 years, and he was saying, yeah, it's been 60 years since we've seen this algae, but we know that it's not there. They have been diving all over the place for 60 years. Right. So it's kind of the same. One of the things is reaching.
Right.
Yeah. Exactly.
Yeah. No, exactly.
And here on newspapers in Chile, there was some rumors about the effect that coronavirus could have in the Antarctic megafauna. But in reality, we have little understanding about this. And after all, there is very limited species that are affected by this virus in the continent itself. So those rumors were really, they lack a little bit of scientific evidence to do more. However, the point is that we don't know, and we should try to avoid any chance of the virus reaching Antarctica, the Antarctic continent anyway. Again, you're talking about teleconnections. We're talking about a relatively small virus as well. So traveling around, would it be something? What are your thinkings about that? Should we be concerned, or? I know we're talking about speculations, obviously, at this point, but it's for
I would be more concerned about us, bringing the virus to Antarctica,
carrying it, right?
Carrying it, yeah. I think it's possible for the virus to get transported. It certainly does. There is some transport documented, local transport, short range, if you will.
Yeah.
Long range, I'm not aware, and it might be difficult to determine just from the genes. In Santiago, for example, we found coronavirus in surfaces, in the air, and it's just genomic debris, you know, like it's not active virus, but it's just some of the genetic material is still there for us to sequence, right? So long range, I would predict that the virus is not going to be active because the virus is an envelope virus. So it has a membrane cover, and that membrane is really not very stable. So I would think that the membrane would stop working properly because the UV radiation and some other pressures, environmental pressures, and the membrane, the envelope is really required for infection. So probably the main vector or carrier for coronavirus to Antarctica is going to be us.
Cool. That's good.
Cool.
Let's keep going.
So we're planning on going this next summer. It's going to be the 57th Antarctic Scientific Expedition that is sponsored by the INACH, by the National Antarctic Institute. And originally, as I mentioned earlier, we were planning on going to Union Glacier down here, but now we're trying to go to this other place, to Lago Telleria Island, further north. It still works for us, at least for this first campaign, right? We're planning on going this summer and the next to have some sort of a point of comparison in terms of stability of the microbial communities. Do we see or do we not see the same taxa being transported? It's, you know, there are some reports from the physical sciences that show that it's really dependent on the current conditions, right? Whether you have a high pressure or a low pressure, or whether you have an El Nino oscillation event or not. It's relevant in terms of the amount of air that is going to be transported there, and therefore particles and the whole deal, right? So we really want to do it in 2 years, and we're probably gonna be taking samples in 2 sites in the islands, and one here is still within the polar circle, but not as far down as the Union Glacier. We're going to be collecting air samples for DNA sequencing, of course. The same samples can be used for microbial culture, so that we can look at the functions and the genes and the phenotype really of these microbes. And we're going to take air samples also for profiling the organic pollutants that we can find along with soil samples and plants. So that back in the lab we can take only the soil that is in direct contact with the roots and things like that, and perform experiments under controlled conditions and so on. So as I said, under this project, we've never been to Antarctica. We're gonna go in December or January. So the next thing was, why don't we go to the Andes Mountains, right? And really we wanted to calibrate or to test the equipment that we have for the air sampling. And really the Andes offers similar conditions to Antarctica in terms of sub-zero temperatures. Of course, altitude, it's different. We were at 3,000 meters above sea level here. But the good thing is that at that altitude here, the density of microbes is predicted to be similar to the one at sea level in Antarctica, right? So it helps to calibrate the equipment. And there's almost no vegetation, which means that we're likely not getting any interference from local sources other than the soil that you can see in this video and so on. So, and we wanted to do that with these 2 equipments. The Coriolis Micro is for the microbial component, and this MCV high volt particle sampler is for particles and aerosols. We were trying different buffers, collection buffers under different temperatures and evaporation conditions, right? For this, we went to this place really near to the border with Argentina. Most of us are based in Santiago. Some of us are based in the south, in Talca and other parts, in Valparaiso as well, over here. But the group in Santiago, we went to this place over here. It's about a 2.5-hour trip, really close to the border with Argentina, and there we set up all the equipment to perform the testing, right? And we wanted to test first and foremost the collection buffer for the microbes, right? Can we use PBS, for example? PBS is probably going to freeze under Antarctic conditions, so we supplemented PBS with 2% glycerol, and we also use PBS with 20% RNA later, which is a very well-known preservative of nucleic acids. It works well for RNA and DNA. And we also tried DNA and RNA shield under different conditions, 2, 4, and 6 hours collection hours at maximum speed. That would be 300 liters per minute, liters of air per minute, right? And we also secure samples for flow cytometry, so we can have an idea of the absolute number of samples that we can get for or by liter or some volume measure, right? And it was a really good trip. So here is our rig. We have some of the equipment here up and running. This plastic container is essentially our laboratory, and you can see a number of things here. And these are the 4 yahoos that went to the area, to the mountains. And it really was. I mean, we made a lot of mistakes, and it was very good because we want to avoid mistakes while we're in Antarctica, and we want to make all the mistakes right now. So actually, we have a better picture of what's going to be deploying all this equipment and gear in Antarctica in December or January. So our project Polarix is committed to training and outreach. We really want to motivate and train the new generation of Antarctic scientists in Chile. And we do so by training, formal training and outreach. We have, or the members of the team are associated with master's and PhD programs in bioinformatics, in microbiology, in genomics, conservation medicine, climatology, and you name it. It's a very large group, and we're all related to or associated, I want to say, to different programs, postgraduate and graduate programs. We have a website that you can visit. It's www.polarix.org or org. We have a show. One of the members works with another very well-known group, the Centro Ideal, in this outreach, I want to say, show. It's of course it's streaming. It's a video podcast. We have a YouTube channel, and of course you can reach us as well in social media, specifically in Twitter. This is the team, right? So Beatriz and I are in charge of the microbial and the viral part of the project. Marco and Jorge from Talca are in charge of assessing the impact of pollutants on Antarctic plants. Nicolas and Dennis, they work in climatology, meteorology, and modeling. So they are in charge of the transport pathways in a changing climate, of the simulations and all that. Ignacio works in metabolic engineering in Universidad Andrés Bello, and he's in charge of the Phenotype-genotype part of the project. And Cristobal is associate professor at Universidad Mayor here in Santiago, and he works. He's been working for over a decade in pollutants, especially in Antarctica. So he's actually leading the origin of organic pollutants in Antarctica part of the project. We have a great team of students and postdocs from different institutions: Universidad de Chile, Universidad Mayor, Universidad Andrés Bello. And we also have a really large group of multidisciplinary, really group of national and international researchers in the States and Europe and Africa and Oceania, and of course in Chile, many of them from Chile as well. And we're really recruiting. You know, we want you. If you feel like you like to work in the project, whether you're from Chile or from abroad, feel free to visit the website. This is the website. At the moment, it's only in Spanish, but it's going to be in English as well in a few weeks. Or simply drop us a line at the email here, polarics@polarics.org. And we're actually actively recruiting. Cristobal has 2 positions, fully funded both, one PhD position and one postdoc position at Universidad Mayor. So if you feel like working in transport of pollutants to Antarctica, it's something that you'd like to do, please contact Cristobal at this email address right here. So some acknowledgements: the government agencies that provide funds for this research, the Instituto Antártico Chileno for logistic support to go to Antarctica and come back from Antarctica as well, and the many institutions that are associated to this project. So with that, I need to thank as well the Escuela de Montaña of the Chilean Army that allowed us to use their facilities right here in the mountains to calibrate the equipment and everything, specifically to these people, Sergeant Vergara, Corporal Quesada, Captain Jimenez, and Commander Olea. They kindly provided shelter, you know, room on board, and good conversations, and all the goodwill for us to work here in this place. So with that, thank you, and I'm happy to take any questions.
Great, Eduardo, thanks. Thanks a lot for that. It was very, very complete. We went from the continent to Antarctica and then back and then go back again. So we went from the air, water, etc. We have a couple of questions still. So there was one that was from the beginning of your presentation, but I guess it's also it is related to the plans itself of the first few projects that you were involved, but it's also related to the selection of sites, because they first of all ask you, like, did you knew beforehand that you were going to find these species, in this case plants, which we have sort of more of information of plants, but I think we can extend that to where to find the bacteria. How do you plan that without being there, or with just so few projects that have been working there?
Right. So for the work we've done before, we were tipped by papers and other researchers, essentially, right? And we knew that in the APECS Peninsula, there were many stable populations of these plants. I've been there twice already, and some of the colonies are not as stable as some papers show. But still we have six sampling sites, and some of those are very stable. The ones that vary the most year to year are the ones that are in proximity to animal colonies, right? Birds and marine mammals and things like that. Regarding the microbes, so for as of now, it's just convenience site sampling, right? Trying to use to use the platform that the National Antarctic Institute provides, and going to places where you have some sort of a preconceived idea of where you can find something. That's more of the culture dependent part that I show you. For this project, for Polarics, we want to do it the other way around. So we selected the samples, the sampling sites, I'd say, in terms of where the air masses hit the peninsula, right? So there's a huge component that goes to Eastern Antarctica. Unfortunately, we don't have good access to that part of the peninsula, but we can go all the way down to Union Glacier, and that's really good because a lot of particles can reach Union Glacier. And then you have the maritime Antarctica, all the islands on the western side of the peninsula, and there's a fair amount of particles from from Patagonia, especially going to those places. And within the islands, we try to select sampling points that are far away, and hopefully they also harbor some sort of plant communities, right? The ideal sampling site for us would be a coastal site where you can find the Champsia antarctica and Colobanthus quitensis, both plants. And from the modeling perspective, it also receives a lot of airflow from Patagonia. That would be the ideal, right? So for the Polarics project, we kind of combined what the National Antarctic Institute had to offer in terms of bases and places we could visit, and the modeling, the air modeling that is available to us.
Mm-hmm. So here we have another question. Leah is asking, can you tell us more about the robot that you want to use for sampling air in Antarctica?
The machine, right? The equipment, right?
Yeah.
So it's really a vacuum cleaner, a glorified vacuum cleaner, right? Essentially, you need to put it at some height so that you avoid local influences, right? And for Antarctica, we're thinking about 2 meters or above, right? And the good thing about it is that it's very fast. So at maximum speed, it can capture 300 liters of air per minute, right? And you can use the machine up to continuously for up to 6 hours, right? So that's a lot of air. Of course, it's a cyclonic device, so you have essentially a cone where you have a buffer, the collection buffer, where the microbes are going to be trapped, if you will, right? Or particles in general, right? Going to be trapped there. But just the cyclonic movement of air within the cone kind of heats up a little bit the solution. And you see a lot of evaporation and even re-aerosolization of the buffers. So you got to keep an eye on that, and that's why you have some sort of peristaltic pump to compensate for evaporation and all of these things. Of course, all of the buffers are filtered and nucleic acid free and ready to work. Besides that, we use soil controls, surrounding soil as controls, and we also have we use one of these collection cones, but we don't put it in the machine, or we put a second machine. We just put the cone there with the liquid, with the buffer, right? But we actually don't capture any air, and that's another control that we use so that we can tell whether the microbes we're seeing are local because they were locally in the air, right? Or transported, right? That's essentially how we do it. And then regarding the provenance or the origin of the air masses that we collect, that's essentially satellite models, high-sat, back trajectories, and also forward simulations that Nicolas and Denise are doing right now. So.
Okay. Another question is that how would you, because there is pollutants coming from the stations as well, how would you differentiate those contaminants that are already from the Antarctic base research stations than those that are actually coming? Well, I guess those also come from the continent, but right. There is active 24/7, the whole year round basis. So
right.
How do you account for that on the project as well?
Right. So there are actually 2 sources, local sources. One is what you mentioned. The Antarctic bases are actually polluting the environment because they work on diesel, on oil, right, 24/7, and that's a huge source of I'd say contamination to our experiments. It's pollution in general, but contamination in terms of what we want to see, right? And the other source is it's melting glaciers and melting ice, really, because a lot of what's being transported and deposited in the Antarctic continent goes to the ice, right? And then, you know, season after season, that builds up, right? And when it thaws, you know, you can release those, and really the equipment is so sensitive that you can pick up those signals, right? I guess it's also long range transport, but not from that point in time, right? It's more archival, right? Some contamination or pollutants. So you have two sources of potential bias or contamination to your experiment. And what you do is that you don't use the Coriolis for that. You use some high volume sampler that has a system where, well, first of all, you just go like 100 meters or so to a high point away from the base, right? That's one thing. Or you simply take some sort of a generator with you and a really long extension cord and up to 100 meters or so, and then you set up your sampling point. That's one, and the other one is you use, I don't know how to say it in English. It's called a veleta. It's essentially something that blocks the airflow from a different point. So you actually configure the cardinal point where you want to capture the air from, right? And then if air starts coming from some other place, the equipment shuts down, right? So that it doesn't get into the filter. That's essentially how you control that. It's probably not perfect, right?
Yeah.
But there are studies that have looked at traces, isotopic marks, and things like that. And most of what you capture, you can tell it's not local. It's long range transport. So.
Yeah. Okay. Cool. Okay, so we're going to move. We're running out of time, but I really want you to address this one because you have had a great and fructiferous career, and you're pretty young, and you're about to no longer be a young polar scientist already because you already have your lab and you already have your whole network, etc. But it's really not the reality of several also brilliant colleagues that today with all the higher competition for positions at the universities and the lack of openings on universities as well. They're just struggling around. And do you have any final advice for those young scientists? Because most of our people that is hearing you are young scientists that are listening, and they are also just starting their careers. So maybe some early decisions that could take them that might benefit them later in the career.
Right. I would say from my point of view, most of what we do as scientists is communication, and sometimes we're not trained as good communicators, right? And that's true for mentor-mentee relationships and connecting with other researchers. I mean, even APECS, right? It's a good platform to connect with people, and I think that's really key because you could start working in a question that is really niche and really unique, and you like it a lot, but you really need to build a network with other researchers, right? So part of what we do in the lab is actually just seminars, outreach, workshops in English and Spanish here in Chile and abroad, and try to connect with people, and from those interactions you start getting up more opportunities, not only for getting a job, but maybe collaborate with somebody else on a project. I'd say developing your unique perspective to a problem, and then connecting with people with other tool set. It's pretty good. In my case, it's genomics and bioinformatics, right? So and I have my biological model, if you will, which is microbes or microbial communities, right? And you can really apply the same methods to different questions, and you can really work on method development and testing new methods and applying those methods to new questions in different systems, right? So for me, it's just environment, but for other people, it's animals, animal health, One Health, Human Health, things like that, right? I think you need to develop a toolkit that is going to allow you to interact and to be useful to other people, so that other people can be useful for you, right? And then networks and a lot of finger crossing, and you go from there, right?
Yeah. All right. Well, Eduardo, I want to thanks so much for having you here today. It's a privilege, really, and especially in this very important week for those that we work and we never want to stop working on Antarctica. You guys can see if you want to follow up what is going on with Eduardo, you can follow his Twitter, but as well we can follow how is Polarix going on that Twitter as well. I would like to remind you guys that also the websites where you can find the lab of Eduardo and the Polarix project. And remember that this is one of our activities that is associated with APEC Polar Week, which we still have some activities. I'm not sure if they already closed the photo contest, but there is a few more other things that you can just Google APEC Polar Week and you will find out all those activities as well. Just to remind you that tomorrow we have another session of Breaking the Ice with APEC-Chile. In this case, this session is going to be in Spanish and it's going to be moderated by my wonderful colleague, which is also here on the screen right now, the Vice President Claudia Maturana. And remember that you guys, it's also going to be about microbiology, and we're going to have Dr. Julieta Orlando from the University of Chile and the Genomic of Antarctic Biodiversity group. So remember, you need to register for this first, and you can find those links in our websites, our social media, or the mail. Eduardo, I don't know if you want to have a final word. We're super happy with this, so.
I just want to thank you and the organizers for the opportunity. You know, it's always weird to give a presentation without results, right? So if you give me the opportunity next year or the next, I can show you some specific results. And thank you for the opportunity. Great work.
Yeah, I'm sure you will have it, man. So we'll be super happy to have you here, especially now that we know where's the thing going. So thanks again. Thank you all the team, the technical team behind, and the people from APEC as well. And I'll see you guys. We'll see you guys tomorrow with another exciting talk. Bye-bye.